to introduce Joanna Kacprzyk from University College Dublin, who will now tell us about plant cell death.
Yeah, that's a bit of a change. Thank you so much. Welcome to Plant World. I'm Joanna Kacprzyk. I'm an assistant professor at Program Cell Death. Can you hear me, guys?
Yes, we can.
That's fantastic. I gave a lecture last year without realizing I was on mute, so that's a good start. So my central research question is, how does the plant cell die? And I'm studying a mechanism called programmed cell death, which is a genetically controlled self-destruction pathway. And that was my huge research interest since I was like, I don't know, first year undergrad. It's just so fascinating that there is this red button that if pressed can activate the controlled cell suicide. So usually when I mention programmed cell death, people are more familiar with cell death programs from animal world, so things like apoptosis and its relation to cancer, neurodegeneration, and so on. But PCD processes are just as important, if not more, in the plant life. If you look at the plant, if you think about the plant life, they are sessile, they're not moving away from the hostile environment or attack or anything like that. So they must have evolved unique strategies to cope with these stresses, and PCD is a part of such strategies. But if you look at the plant life, starting from reproductive development and seed formation and the development of the multicellular organism and interactions with environment, PCD is part of these responses. So for example, during seed formation in cereals, there is this part of a seed called endosperm, and endosperm formation relies on programmed cell death. And this is a big deal for us humans as well, considering that 2/3 of human calories consumed by humans worldwide come from endosperm. Mind-blowing number. Likewise, interactions between plant and environment, including abiotic and biotic, so pathogen responses, are mediated through PCD events. Similarly, the end of plant life, so senescence, is finalized by PCD events, so that may have consequences for post-harvest storage durability of things like vegetables and cereals. So in a conclusion, plant life needs cell death, and in order to understand plants and make them happy, we need to understand cellular cell death in plants as well. So this has never been as important as now, perhaps, because we're all facing a grand challenge. We all need to maintain the plant production despite increasingly volatile climate. So while I believe there is no silver bullet to this solution, increasing how well we understand plant programmed cell death, I think is a part of this solution. So how does the plant cell die? Well, we don't know yet, right? We know that part of the problem is that dying cells are often buried within the bulk of the tissue, so that makes them somewhat hard to study, hard to sample. This is changing with better, more advanced technologies as well. We also won't find the homologs of some of the key apoptotic genes from animals in the plant genomes. However, when we look at the events happening in the plant cells during PCD, we'll notice that certain events are actually conserved or appear conserved between plant and animal kingdoms. So you'll see similar pattern of nuclear degradation, you'll see mitochondrial involvement, ROS, reactive oxygen species, calcium fluxes being regulators of the process. So it might be that there is a similar play between plant and animal cell deaths, but just different actors in the role. So how do we identify these actors? The system I'm using in my lab is the cell suspension culture. It's a very well-established system for PCD studies in animals. The species I'm using is Arabidopsis thaliana for non-plant people, which is most of you probably. Arabidopsis thaliana is literally like lab mice for plant scientists. So we have loads of resources and reagents developed for the species especially. And these are the cell suspension culture. It's basically a flask, liquid medium. That means it's very easy to add chemical modulators to the flask to induce programmed cell death. You can also treat them with heat or something like that to induce PCD, but you can also add drugs easily that are blocking or modulating PCD process. What's also pretty cool about the system is that you can easily withdraw a small sample of these cells, maybe like 20 microliters or so, and you can see what's the time course of the PCD occurring after you've induced it. So pretty much you can pinpoint the timing before the cells are actually dead, but you can presume they already committed at this stage to the entry to the PCD. This is how they look like under the microscope. 2 PCD cells from Arabidopsis. They have this morphology where the protoplast is retracted away from the cell wall, it's shrinking, and there is this nice visible gap between the protoplast and the cell wall. So as you can see, something very easily recognizable under the microscope, just regular light microscope, no specialized equipment required. So how I'm using the cell culture system in my lab. My first student, Rory Burke, he's using it to study the transcriptomics of PCD in plants. He's using different PCD inducers, for example, salicylic acid. So this is a phytohormone involved in pathogen responses. He's using heat, so abiotic stress. He's also diluting them so much they're basically starting to die because they can't talk to each other because they cannot exchange survival signals. And he's also blocking the program cell death using things like 3MA, which is, we think, inhibiting the mitochondrial swelling during PCD, and lanthanum chloride, which is a modulator of calcium signaling, or he's also adding back these survival signals to the medium. He's using different combination of these treatments and harvesting the RNA at the point when they are not dead yet, but we can presume they're already committed to the PCD entry. And after the RNA-seq has been done, Rory is doing differential gene expression analysis. So basically he wants to figure out which genes are either switched on or switched off when the cell decides to die. So loads of death, no death comparisons allowed Rory to identify a bunch of candidate genes. So these are the genes which were commonly switched on or switched off when the cells were dying. This is an upset plot. Well, the name maybe, it is a bit upsetting looking at it. It's very hard to figure out what's going on. But believe me, if you try to present it on the Venn diagram, it would be even worse. But basically here, as you can see, there is quite a lot of genes that appear in, at least 4 or 5 of the comparisons he's making. So then the advantage of working with Arabidopsis thaliana is that such a well-established species in plant science, there are beautiful genetic resources. So Rory could order knockouts or knockdowns in the genes that he isolated, and now he's doing some functional validation. So basically he's looking at different aspects of plant program cell death in these mutant lines to see whether they were affected by disruption of a gene. So for example, here we have the wild type leaves, which are already dead, all bleached after being detached, but the mutant leaves do not seem to be senescing yet, so there is a difference. And this is early stage of this project, but I would say watch the space, because I think we should be getting something exciting within the next year or so. Another student of mine, Jo, she's also using the cell suspension culture, but she's studying one organelle and its involvement into plant PCD regulation. It's mitochondria, right? So mitochondria, everyone knows, there is even a meme, mitochondria, the powerhouse of the cell, power horse of the cell. But so they produce this all precious ATP within the cells, but they can also turn into killers if their pro-apoptotic proteins are released from the intermembrane space and sometimes matrix into the cytosol that can trigger a cascade of proteases leading to destruction of the cell. So this is known and well defined in animals. In plants, we know that mitochondria are very likely to be involved in plant PCD regulation. There are different pieces of evidence, but we don't really know the details. And this is Jo's job to figure it out. So what she's doing, she's killing the cells, and then minutes after the cells reach the point of no return, so the PCD was induced, she's separating the mitochondrial and cytosolic fraction. There is a yellow arrow indicates her precious mitochondrial band. And then she's using mass spectrometry to identify the proteins present in cytosolic and mitochondrial fraction and their abundance. So hopefully that will allow her to identify which are the proteins released from mitochondria into the cytosol just when the PCD is induced. So far, so good. So our findings seem to support the devil inside hypothesis that mitochondria are these, it's an insider job, right? The mitochondria are driving the PCD process. She identified the over 100 proteins released from Arabidopsis mitochondria to the cytosol. And excitingly, only 4 of these proteins were not released when we blocked the PCD using a chemical agent. And what is really, really exciting is that one of these proteins has an MDM2 domain, which is known to be common in proteins regulating the programmed cell death in animal kingdom as well. So loads of validation ahead of Jo in her project, but hopefully exciting results. So this is devil inside, but we also have a devil outside hypothesis based on the results. So she noticed that there are some proteins from the extracellular space, from the plant apoplast. 9 of them seem to be imported back into the cytosol when the programmed cell death is induced. So we think that they might be actually executing the PCD process. Interestingly, these proteins, they seem to be autolytic in nature. There are peptidases, ribonucleases, endonucleases there. So these guys could be damaging in the cytosol of healthy living cells. So they might be kept in the apoplast, but they're also it would make sense for them to be reimported to execute the cell, the PCD after the trigger. And it's not a totally new idea. That was shown in another species for just one protease that is reimported from the apoplast back to the cytosol for PCD execution. Although our data suggests that this might be actually a more common mechanism than initially thought. Okay, so again, watch this space. Hopefully next year we'll have a little bit more to say. The conclusions are, science of death is really cool. I think you cannot understand life without understanding cellular death. So I love it personally. It looks like well-established models like Arabidopsis cell suspension culture work well with the state-of-the-art technology, like good mass spec, RNA sequencing. We identified interesting targets already. Validation awaits. We were slowed down by COVID a little bit. Ireland has very big, well, kind of severe restrictions linked to COVID, but now hopefully we'll go full steam ahead and we'll bring more data to maybe next meeting. Big thanks to people who, well, all the funders, of course. Without them, that wouldn't be possible. Team and collaborators, and if you want to chat about life, death, or plant cell, or life in general, you can always contact me at the email provided. Was that okay? Was that 10 minutes? I think I'm done.
Yeah, timing was perfect. Thank you very much.
I was freaking out about timing.
I just have a quick question. So you said you were able to identify some candidate genes in the right? Is it kind of conserved among different plants?
Well, it looks like they are conserved, yes. So this is exciting. Although, you know, it's I'll be sleeping better after we validate that these genes have anything to do with PCD processes in whole plants, because this is one of the problems with the study. You know, a reviewer too will always say, well, these are cells. What about whole plant? I think my line of defense, and hopefully I'm right, is that it's such a conserved core mechanism present in all cells that, you know, maybe undifferentiated simple cell system is actually a good way to study it. So yeah, they are conserved, yeah.
And so then the next step would be to knock out some of these genes in
in plants, and maybe even look beyond Arabidopsis, I would say, maybe some grasses at least.
And we have one more question from Joanna. So is the programmed cell death in plants also driven by climate or climate change?
Well, driven. Well, This is a really interesting question. It's almost like you've read our lab's notebook. So program cell death, like, plants also have, in addition to mitochondria, they have chloroplasts, right? And the chloroplasts are also, they're producing sugars and so on, like photosynthesis happens there, but they're also a source of reactive oxygen species within the cell. There's huge evidence that chloroplasts are also modulating program cell death. And if you think about the climate change, you need to, you think about increasing CO2 concentration, more CO2, more photosynthesis. Plants are photosynthesizing like crazy. So there might be more reactive oxygen species within the cell. And the cells might be more likely to they might be more sensitive to PCD triggers. So yeah, definitely. We have some data actually showing that at higher CO2 concentration, the cells are undergoing higher rates of PCD. It's very complex actually, but I would say this is definitely a part of a thing that we need to consider.
Very great. Well, thank you very much again, Joanna, for the.
Cheers, thank you very much.
And
I'll stop sharing now. Thank you.